Welcome to the part of the educational course concentrating on the analysis of cortical layers in the human brain and in that of macaque, monkeys and rats, two of the animal models most commonly used in neuroscience. The numbering convention of cortical layers can be considered arbitrary in the sense that structures classified by some neuroanatomists as two distinct layers are described as a sublamination of a single layer by other researchers. The most widely accepted convention was first postulated by Brodmann in 1909 and is based on his observations that the laminar location of cortical neurons, and more specifically of their cell bodies, is determined during development. Brodmann described a canonical organization of the cortex into 6 layers. Layer 1, the molecular layer, occupies the most superficial parts of the cortex and contains only a few scattered small neuronal cell bodies. Layer 2 is the external granular layer and contains small neurons with roundish, grain-like cell bodies, hence the layer's name. Layer 3 is the external pyramidal layer, and as you can imagine, the cell bodies of neurons in this layer are pyramidal in shape. Layer 4, the inner granular layer, also contains small granular cells, and its thickness varies considerably between the different isocortical regions, reaching the largest width in primary sensory areas. Layer 5 consists mainly of pyramidal cells of large but variable size and is most prominent in areas of the cingulate cortex. Whereas layer 6 is called the multiform layer because it contains cell bodies of different shapes, for example, spindle-like or polygonal. Layers 1 to 3 are the so-called supragranular layers, that is, they are found between the pial surface and layer 4, whereas layers 5 and 6 constitute the infragranular layers. In some areas, sublayers can be defined based on a gradient in the size and/or packing density of the cell bodies they contain. So, for example, although layer 3 is mainly characterized by small and medium-sized pyramidal cells, in some areas very large pyramids are found in the lower third of layer 3, the so-called layer 3C. Now, not all cortical areas display this basic laminar pattern. Cell birth and cell death, apoptosis, which are normal components of brain development, are associated with the creation and development of layers, as well as with their regression. Brodmann defined 2 types of cortex based on whether they presented his 6 canonical layers during both fetal and adult stages. His homogenetic or uniform cortex presents the 6 layers. Heterogenetic cortex can present either less than 6 layers, as does, for example, the hippocampus, or more than 6, and a typical example of this would be the entorhinal cortex. These 2 cortical types, homogenetic cortex and heterogenetic cortex, are what we nowadays call isocortex and allocortex. I mentioned sublamination earlier on, and in the primary visual cortex we have an extreme case of sublamination in layer 4. In the primary motor cortex we find the opposite situation, where layers 3 and 5 become so prominent in adult stages that they completely disperse layer 4 granular cells in the adult brain. And this isocortical variant constitutes the agranular cortex. This schematic drawing of the mesial aspect of a human hemisphere visualizes the ratio between isocortex to, let's call it for now, non-isocortical cerebral surface, where the mesocortex was identified by Brodmann as the portion of the cerebral cortex occupied by areas which present what he considered a transitional laminar pattern between allocortex and isocortex. For example, his area 24, located on the rostral portion of the cingulate gyrus, belongs to the mesocortex. Specifically, it is what we call pro-isocortex. It has 5 clearly developed layers, and the only difference with the neighboring isocortex is that it does not have a layer 4. Now, why is it important to know of these different cortical types? It's because they are differentially vulnerable to disease, and they can help us understand, for example, the sequential changes in the distribution pattern of neurofibrillary tangles during the course of Alzheimer's disease. Bragg identified 6 stages, and in this schematic representation, the increasing density of shading indicates increasing severity of neurofibrillary changes. Stages 1 and 2 show alterations which are virtually confined to a single layer of the transentorhinal region. And the key characteristics of stages 3 and 4 is the severe involvement of the entorhinal and transentorhinal layer pre-alpha, whereas by stages 5 and 6, the destruction of the isocortex is the most prominent. Although most classical architectonic maps are based on the analysis of cytoarchitecture, some neuroanatomists of the time already recognized the value of a multimodal approach to identify cortical borders. This figure by Brodmann now also includes his schematic representation of myelinated fibers in the cortex, and I have adopted his convention of using Roman and Arabic numerals to identify cytoarchitectonic and myeloarchitectonic layers, respectively. As you can see, there is no one-to-one correspondence between the layers revealed by these 2 staining methods, though we do find concordances for certain layers, such as layer 4. It's specifically this outer stripe of Gallagher that is particularly enlarged in the primary visual cortex of primates, where it has been named the Gennari stripe, and I'll come back to this structure later on in my talk. Quantitative in vitro receptor autoradiography is a modern technique enabling the visualization of neurotransmitter binding sites with a high spatial resolution. Here you can see snippets through the human primary visual cortex. The sections they were taken from were processed for the visualization of cell bodies, myelin fibers, or of different receptor types. And color coding of the receptor autoradiographs is such that cold tones stand for low receptor densities and warm colors for high densities. I've transferred the lines demarcating the Gennari stripe from the myelin staining to all other sections. So although receptors also present clear lamination patterns, you can see that they can differ between receptor types, and also that there is not necessarily a one-to-one concordance in the number of layers revealed by each receptor type or by the receptors or the classical staining methods. Receptor autoradiography has the advantage of being fully quantifiable, and receptor fingerprints, that is, polar coordinate plots simultaneously depicting the densities of all examined receptor types in an architectonically identified area, reveal a canonical sequence of receptor densities. These 3 exemplary areas show that highest receptor densities are generally found in layers 1 to 3 and lowest ones in layers 5 to 6 in the infragranular layers. The glutamate kinase receptor constitutes a notable exception, since in most areas it's actually present at higher densities in the deeper than in the supragranular layers. Now, this canonical sequence does not mean that a given receptor type presents the same laminar distribution pattern throughout the entire cortex. The muscarinic-cholinergic M2 receptor is an extreme example with a relatively wide range of laminar distribution patterns. Some areas of the cortex, for example, primary somatosensory cortex, area 3B, or primary auditory area 41, present a single peak with maximal densities located in layer 3. Other areas have two peaks with higher densities in superficial than in deeper layers. And examples of this are the primary motor cortex, area 4, or the primary visual cortex, V1. Now we can also find areas with lower densities in the superficial than in the deeper cortical layers, and temporal association areas or the cingulate cortex, area 24, would belong to this category. And finally, some areas also present an intermediate maximum in their receptor profile. For example, somatosensory area 2. Now, moving on to the comparative part of the talk, here you see lateral views of the left hemisphere of a human, a macaque monkey, and a rat brain. The 1 centimeter scale bar applies to all three images. The differences in size between these three brains are obvious and not important for this course. And the fact that the rat brain is lissencephalic, that is, its cerebral surface is smooth, whereas in the human and macaque brain the cerebral surface is convoluted, isn't relevant either. What is important is the fact that the human brain has a proportionately much larger isocortex than any other species. Here again, the human brain, and next to it that of a primitive insectivore, the hedgehog, a basal primate, the prosimian galago, and an old world monkey, the genon. Macaques are also old world monkeys, and their ratio between isocortical to non-isocortical cerebral surface is comparable to that of the genon. These are cell body stained coronal sections through a human, a macaque monkey, and a rat hemisphere. Note that here I haven't taken the relative brain sizes into account for visualization purposes. The hippocampus is highlighted by the red frames in all three sections. At a first glance, the hippocampus of humans, macaques, and rats may look quite different because of their different positions in the brain. This is the result of the expansion of the isocortex in the human and the macaque brain, and this is particularly true for the human brain. Thus, all of this developmentally new cortex has literally pushed the hippocampus in primate brains ventrally onto the medial surface of their temporal lobe. When we zoom into the human and rat hippocampi, however, we can see a great deal of similarity, although there are some differences as well in cytoarchitecture. In both species, the cornu ammonis region, highlighted in red, warps around the dentate gyrus, highlighted in blue. The granular layer of the dentate gyrus, which contains the cell bodies of the principal cell type in this region, is comparable in width and packing density in both species. However, there are considerable differences in the packing density of the pyramidal layer in the human brain. The cytoarchitecture and also the receptor architecture of the macaque hippocampus very closely resemble that of the human hippocampus. The differences in cytoarchitecture between the rat brain and that of humans or macaques become even more prominent when we move into the isocortex, and I'm now going to show you two examples of it. The first example comes from the primary visual cortex and illustrates why it is called striate cortex in the primate but not in the rodent brain. In the primary visual cortex, there is a band of densely packed myelinated axons running parallel to the cortical surface, the so-called Genari stripe, which corresponds topographically to cytoarchitectonic layer 4A. Layer 4 of rat primary visual cortex is not sublaminated, thus we don't have an A, B or C component. And furthermore, it's obvious that cytoarchitectonic layer 4 in the rat brain does not contain a band of densely packed myelinated axons running parallel to the cortical surface either. In the second example, I want to highlight cytoarchitectonic differences between the two primate brains and that of the rat. I had mentioned that layer 4 is not visible in the human primary cortex at adult stages because it has been invaded by pyramidal cells from layers 3 and 5. Layer 4 can't be seen either in silver cell body stain sections through the macaque primary motor areas, but a layer 4 is clearly visible in the primary motor cortex of the rat brain. Furthermore, layer 5B of the primate cortex contains a specialized type of pyramidal neuron, the so-called Betz cell, but the rat primary motor cortex does not present these gigantic pyramids. These differences in the cytoarchitecture of the primary motor cortex are accompanied by differences in receptor architecture. For example, whereas in the primate brain, the second maximum in the muscarinic M2 receptor profile is considerably lower than the first one, in the rat brain, both maxima reach a comparable height. If we concentrate on the primary somatosensory cortex, we find a comparable cytoarchitecture in all three species. However, the M2 receptor profiles of human and macaque area 3B differ considerably from that of primary somatosensory area PAR1 of the rat. And we also find considerable discrepancies in the laminar distribution pattern of the 5-HT1A receptor. Whereas in most areas of the human and macaque brain, highest densities of this receptor are generally found in layers 1 to 3A, in rats they are located in layers 5 and 6. And why are these laminar differences so important? It has to do with the fact that cortical layers also differ in their connectivity patterns. Layers 5 and 6 are the source of all cortical output. They are also the layers in the human primary visual cortex, for example, with the lowest synaptic and overall receptor densities. Conversely, layer 4, which is the main target of thalamic projections, is also the layer with the highest synaptic and overall receptor densities. So, concluding, neurotransmitters and their receptors are key molecules in signal transfer and thus bridge the gap between the structural and functional segregation of the brain. Humans, non-human primates, and rodents differ in their cyto and receptor architecture, not just at the regional, but also at the laminar level. And therefore, selection of the correct animal experimental model is of utmost importance in neuroscientific research. And with this, I would like to finish. Thank you for your attention, as well as my team for their support and hard work, and Catherine and the funding agencies.